anymore. Who wants to get hurt every time that you pick a fruit? And we kind of expand on the existing genetic variation in the wild and in the early domesticated species to create all of the varietal diversity that fits the different cultural preferences around the world. There's my eggplant slide. So now I want to quickly digress and talk a little bit about Nikolai Vavilov. Has anyone heard of Vavilov before?
I know his son.
You know his son?
Yeah.
How old is his son?
He died a few years ago. He was a physicist.
Oh, okay.
And Armin Tucker's on. Was very close to him. Never got over it that.
Yeah, it's tragic. Vavilov, basically before the Green Revolution, Vavilov had probably the coolest job in the world. He got to travel all over the world and collect different crops that were used in South America, in different parts of Africa, all throughout Asia and Europe. And then he took them back to Russia and he grew them together and was able to look at their phenotypes and say, well, these are related, but came from different places, but they have similar morphology, similar features, and they differ in similar ways from their wild relatives. So he actually started the first germplasm bank, and we're still actually fighting to preserve it. He and 11 of his staff from that germplasm bank ended up actually dying to protect the seeds during the war, and he ended up dying in prison. But some of the things that he really helped further our understanding of are okay. Of course. Okay, we're back. We're back.
It's just set to go to sleep.
Oh, okay. Sorry. I'll just have to keep moving faster. So he really helped develop our concept of that there are domestication centers across the world. And he also founded the law of homologous series of variation, which is basically where related species or subspecies have similar potential for evolution of forms and metabolites. And so basically you can say these are related. So if this species can have red fruit, then maybe this species can also have red fruit. And so you can actually start to look at, you can make predictions, and you can start to look at patterns within the processes of domestication. So now I'm going to talk about what I'm interested in. I'm interested broadly in crop evolutionary history, in the genomics, genetics, metabolites, and ethnobotany all about our crops. And I worked on this review with Michael Fruganen, who will be presenting later today, where we sort of pulled apart the domestication process into early onset of domestication and then diversification processes, which I'm calling here stages 1 as onset of domestication and stages 2 through 4 as diversification processes. And so throughout, as we move away from the wild phenotypes and start to domesticate and increasingly select upon desirable traits in our crops, these different patterns start to occur. And I want to ask a lot of different questions about the different stages of domestication, such as what was under conscious versus unconscious selection, or what were people using the plants for and what was desirable when people wanted to amplify desirable alleles that represent traits, like maybe to make something tasty. What was it that people were using the plants for that helped lead them on a certain evolutionary trajectory? What were the environmental challenges, and how did human technology and the plant genome respond to them? So when you have your crop plant and you have to move to another environment or a new culture adopts it, what are the challenges that come with that? And what are the traits of maybe underdog land races that we can exploit? So these different stages of domestication and diversification frequently can be encompassed by different patterns of traits that people are selecting on. So early on you might see larger seeds in the archaeological record that distinguish the early cultivars from the wild forms. And then later in domestication, maybe you see more pigment changes or flavor changes. And in stage 3, when people are moving the crops further away from their native range, maybe you see changes in flowering time or life cycle or dwarf plants forming. And then in stage 4, increased yield, different stress tolerance, these very targeted traits are often under selection. And this is not just within one species. This is sort of across the board for seed crops or fruit crops. You often see the same traits under selection. So if we relate this back to Vavilov and the homologous series of variation, I'm really curious what variation, maybe in multiple species or multiple varieties, was exploited at each of these stages, and what are the patterns that we can glean from those. So this is one reason that I'm really enthusiastic to be a part of the Peruvian lab team, because there's so many projects in Michael's lab that really deal with crop adaptation and just evolution in so many senses that I can start to make cross-species comparisons. So I'm going to quickly tell you a little bit about the projects that we have in our lab that I feel very happy to learn from. This is a picture of a terrace agricultural system. So one of the questions that we're asking is, how did the genome of rice evolve to adapt to the terrace environment instead of the lowland environment where it was domesticated in? This picture of Arabidopsis, this one's not flowering, this one's flowering. So people in the lab study what is the molecular basis of being able to adjust your flowering time depending on the different climate or environment that you grow in. People study salinity tolerance, salt tolerance, like a graduate student in our lab, Ines Pires, and how the genome can vary to be salt sensitive or salt tolerant. We study rice environmental response. This is a postdoc in our lab, Anne Plessis. She's in the audience here. She basically can take rice varieties that are in different environments and have a long history of growing in different environments and compare how their genes are being expressed differently and what that means in terms of their relationship to the environment. We also, people in the lab have a lot of experience putting together models for understanding the domestication history of our crops better. We also have this really cool date project called 100 Dates that Michael will talk about later, where we can look at the domestication history and the diversity and how that's changed, and sort of how variation in date palm exists across the landscape of where dates are cultivated. And we can look at the molecular basis of desirable traits like fruit color or sweetness that could be useful for the market. So sort of all of my questions from the different stages of domestication fit in to some project in the lab. And now my specific project is on African rice, as I mentioned. And so because we have so much experience studying Asian rice, it's really neat to throw in another species. These are closely related species, so I can start to ask all these questions that relate back to Vavilov or are about, and that are about parallelism. And I can explore possible integration between these species and the different adoption of practices, because these two species, even though they're closely related, they have very different histories of use. Asian rice, or Oryza sativa, was domesticated about 9,000 years ago in China. And Oryza glaberrima, African rice, was domesticated maybe as early as 3,500 years ago, 3,000 years ago, in the Niger River Delta, which is actually an upland rain-fed environment. And then it was subsequently brought into different areas that were coastal. I'm going to give you a little bit of history on why I became initially interested in African rice, just because It's too important to pass up. I read this book called Black Rice by Judith Carney, who's a geography professor at UCLA, and she basically has been spending her life trying to showcase that during the development of the United States agricultural, profitable, cash crop system in the Carolinas, where people were growing rice, the slaves whose labor was exploited to create that system, when certain slaves that were from rice-growing areas in West Africa, when they were runaways, there was a higher price for the reward if they were to be captured. And this meant that people were actually really, really dependent upon their native knowledge from growing African rice in West Africa. And people exploited that knowledge and used all of the technology that had developed in Africa to transform the landscape in the Carolinas to make it a profitable agricultural system. And it wasn't until the 1970s that West African technology was really credited for this major economic breakthrough for the U.S. So here's an example of the way that the rice fields are diked in South Carolina, and here's an example that's very similar in Gambia. So, as I said, African rice is domesticated. It was domesticated inland and then brought into these environments that are flooded by coastal water. So there's a lot of salt, and most crops don't grow well in salty conditions. So people consciously selected for varieties that were more salt tolerant. At the same time, people were, desalinating the soil, transplanting the seedlings, finding the best time to transplant the seedlings, diking the fields to collect more rainwater and hold it there, and develop this really advanced technology to be able to mitigate salt toxicity. So I want to look at this situation, ask all my questions, and so what I did was sampled 100. I collected accessions from IRRI, the International Rice Research Institute, that has all these land races of African rice stored. So 126 land races that sort of evenly represent this upland environment and this coastal environment that were collected back in the 1970s, and started exploring them for different trait variation, and started exploring their genomes. Just a little bit more about African rice. It's really prized for having a lot of tolerance to what Asian rice is frequently sensitive to. So iron toxicity, nematodes, drought, soil acidity, and these have really been shown. But salt tolerance, which has been documented by Judith Carney as being something that people were consciously selecting for, and sort of from the ethnobotanical world has been documented, but nobody had actually shown if African rice was salt tolerant. So that was one of my first questions. So at IRRI, which is at the International Rice Research Institute in the Philippines, people have frequently used this early seedling salt tolerance test, where they'll plant the rice plants in these little trays. After about a week, they'll add a saline solution, and then you see which ones die. So I did that test in order to make the African rice results comparable to the Asian rice results, but also taking into account that farmers often transplant at a late seedling stage. We delayed the test for a week and then more rapidly increased the salinity levels in the solution. And so we actually got two pretty independent measures of that would suggest that maybe at certain stages there's different accessions and different levels of tolerance. So So we used this salt evaluation score, which is 1 through 9 score. 9 means you're dead, 1 means you're like, perky and alive and never even noticed that there was salt in the water. And then we also measured the 4th leaf sodium-potassium ratios. And so that was for the normal test, and for this delayed salinization test, we also measured, 1 through 9, the salt evaluation score, and then also measured the sodium-potassium ratios, but for the entire chute, because more of the plants were still alive, but in the process of dying, so we could take advantage of having a larger sample of tissue to represent the test. And what was interesting was there was correlation between some of these tests, but not among all of the tests, suggesting that these are independent traits that we're picking up. And I won't go through all of these details of the results, but one thing that was really neat is a third of the accessions, after putting them in EC18, which is really, really salty solution, 33 of the plants, of the accessions out of 126, still survived. And a number of them, many of them were fairly tolerant, and some were still very sensitive. So we actually saved the surviving plants at Erie for breeding programs, which I think is really cool. But one thing that was neat was all of the salt-tolerant plants, guess where they were from, the upland or the upland rainfed or the salty environment areas. Yeah, everyone's going, yeah, of course. So the salt-tolerant plants were from the coastal areas, but the coastal areas also were growing a lot of salt-sensitive plants. And so you might have different areas in your field where you don't have a lot of salinity problems, and you can still grow some of the more prized accessions of African rice. So this is cool. So now moving on into what we're doing with the genome. Each of these accessions, well, not all of the 126, but 101 of them, I did whole genome resequencing on, aiming for a 10x coverage. This is a picture of our sequencer. And then we started to explore the variation in different regions of the genome across those accessions. And Inesh, who studies Asian rice salt tolerance in the lab, lent me her genomes from 56 accessions. And so we could actually start to compare the variation between the two species and look for parallelism. So I just want to, this is a circus plot. It's like the thing that everyone has to publish in a study about genomes now. You'll always see a circus plot. So this is basically, this pointer doesn't really work. So you've got 3 different layers of data that are plotted in the same place. Each of these sections each of these segments is a chromosome. And where I have arrows are regions where there's reduced nucleotide diversity. So you sort of see, here it's really evident, there's higher levels of diversity and then sort of it trails down. And that's a pattern that is not actually in all species, but it is in Asian rice. So you see very similar patterns of where diversity is in the genome between African rice and Asian rice, suggesting that they're very similar. Also, what's kind of noteworthy is we had a nucleotide diversity value of 0.004, which, just to put this in the framework, for Asian rice, it's 0.0035 overall. So that was substantial genetic diversity, and only slightly lower than the estimates for the wild progenitor of Asian rice, Oryza rufipogon, which was 0.0058 is sort of the high estimate of nucleotide diversity. And there are, even in some subspecies of Asian rice, there are entire, like, long tracts, long pieces of chromosomes where there's no diversity whatsoever. The genome's been completely fixed. So all the varieties within a subspecies are going to look pretty similar for certain areas of the genome. And we don't see any major reductions in diversity in African rice. So one thing I wanted to initially do was to sort of look for thinking that I would also pick up areas that have no diversity, I thought, well, I'll look at the areas that were fixed and see, which is what we call a strong selection suite, and I would look at what are the genes that are in those regions. But that requires a little bit more work right now. I'm just in the middle of just really starting these analyses. So I started to first look at the regions that had these interesting patterns in LD or in sort of in different ways that you can interpret if the genome is varying in an expected way. And there are some really cool genes that are under these big peaks in the Sotos plot that are related to maybe to environmental adaptation, something that is about detoxification. We know that African rice is really good at thriving in high iron soils. So maybe this particular mate gene plays a role in that. Out of 76 mate genes, maybe this particular one is interesting and people should characterize it, and maybe it can be useful in breeding programs for growing rice in high iron soils. I'm just, this is the beauty of just giving a talk, is I can throw out all these hypotheses about what's going on to you. So there are some really interesting patterns that we might be able to relate to how African rice, when it was brought into coastal West Africa, how it evolved to adapt to that environment and to the different cultural preferences. Maybe also there were some genes that were related to increased selfing. So actually some of the physical properties of what allows a rice flower to open and be open pollinated versus to remain closed. There's a difference between the wild and the domesticate, and there are candidate genes for that have already come up under these major peaks of LD. So we can also take those candidate genes and look at their allelic variation. So are there two sort of main versions of those genes that we see across the 101 accessions? And it turns out that there often are. You can see here interesting geographic patterns where so each of these leaves, this is the version of a gene of this auxin response factor in each accession. And you can see that these accessions that are very similar to each other genetically all are from around the Casamance River. So, maybe there's some reason for that, and we can start to explore these patterns. So now I'm going to show you some of the results, some of the preliminary results about where we were exploring parallelism between Asian rice and African rice, and relating it back to the different stages of domestication. So this is just sort of to give you a flavor of what we can test with all of these genomes. So I'm going to talk a little bit about pigment change, red rice versus white rice, which is considered a very early trait in domestication history, because if you wanted to see if there was, if you had harvested a lot of rice and you wanted to see if there were like rocks in your rice, has anyone ever found a stone in their rice when they're trying to make rice? You have to wash it and look through it. This is something most people have to do. So if you have red rice, it's harder to see if there is some sort of contaminant or something like a rock in your rice. But if you have white rice, it's much easier to see. So this is theoretically something that would be selected early on in domestication. And the other example I'm going to talk about is shattering and non-shattering. So shattering is the term when a plant grows, it has its seeds, and then it's going to drop all of its seeds on the ground. And they'll be carried, swept by the fur of animals and carried somewhere else, or by the wind, or whatever, or by whatever seed disperser mechanism. But where the seeds are not retained on the plant, they're dropped. So wild rice drops its seeds. One of the first traits in domesticating rice in both African rice and Asian rice is the plants will retain their seeds, because it's easier to harvest if the seeds are still on the plant and not on the ground. So I'm going to talk about those two examples, because they're something that would be a trait in both rice species. So here is a really nice study that was done also from the Prugen lab lineage. Kevin Olson and Brianna Gross did this study on the RC gene, which is the gene that is in the pathway that makes a red rice versus white rice. And we can actually see here, I'm going to walk over again. This is a tree of the different versions of this RC gene, that there are several alleles, several mutations that distort or knock out the function of RC in Asian rice. And I know it's hard to read, just bear with me. In African rice, the same gene was affected, but the mutations, the particular sites where there are differences that disrupted the gene function are different. So we can also use this to say how many times was this independently selected. But it's really neat that the same, out of all the genes in the pathway that could make white rice versus red rice, the same gene gets hit in both species. It's a beautiful example of parallelism. Of course, the actual mutations underlying that are not the same. So there's our little parallel stamp. And here are some other examples. BADH2 is a gene that confers, that when you knock it out, you disrupt its function. You end up with a more aromatic rice, so like basmati is aromatic. And this is even widely, you see the same gene being affected in several different species. Waxy is a gene that if you were to disrupt its function, you get a sticky rice. Has anyone ever had sticky rice before? It's really good, huh? Yeah. In sushi? That's a low amylose content rice, but there are stickier rices out there too. Yeah, so actually there's sticky corn and there's sticky sorghum. There's lots of sticky grain species because the same gene, when you knock it out, you get a sticky phenotype. And then on the left I have images of SH1, which is like the main locus for, if it's functional, the plant will shatter, the seeds will drop, and if it's not functional, the plant retains the seeds. There are several other shattering genes, but this is really the main one that's been shown to be responsible for shattering versus non-shattering in sorghum on the top, maize in the middle, and Asian rice on the bottom. So that's 3 grass species. You'd figure, you know, parallelism is really, really common, not only in the grasses, but broadly. So let's explore shattering in African rice. This is from the greenhouse. There's still a bit of a mess I had to clean up because Oryza bartii, the wild ancestor of African rice, shatters. It loses its seeds. So they're all on the ground and I have to sweep them up. And in the domesticate, you see varying panicle architecture, but usually the plants keep their seeds. And one thing, so this is this is called a coverage breadth analysis, where each of the columns that you see are different exons, the different parts of the gene for SH1, so for the shattering gene. And on the left are different accessions. There's Asian rice accessions on the top. They start with sample underbar, and then my African rice accessions start with Erie. And you can see that, so just going back here because it's bigger, the gene doesn't have to be completely deleted in Asian rice to be non-functional. There's actually one SNP. There's a T to G change. I mean, take a change upstream, really far upstream of the gene, that will actually make it so you don't get an SH1 protein. So the gene's still present in Asian rice, it's just not functioning. So what other people have noticed and what I noticed is in many of my African rice accessions, do you see a bunch of zeros there? The gene's completely gone. Completely gone. In fact, there's been a 45 kilobase deletion in this region, which people have suggested means that you don't get shattering at all. And in fact, you get less threshability because you've completely ablated that abscission layer that would normally allow the plant to shatter. But when we compare that back to whether the plants that have the mutation or don't, if there's a correlation between having the gene or not and shattering, we see that there is no pattern whatsoever that suggests that this locus is actually responsible for shattering. So this says very low shattering and low shattering, and you can see very low and low are interspersed, and the accessions that have the gene or don't have the gene vary with no correspondence to whether they shatter or not. In fact, there are some African rice varieties that still shatter, and there is no obvious pattern about whether they have this locus or not. So this is one of our examples where we don't see parallelism. And so this whole mechanism that would have allowed people to really harvest and use this crop to harvest the seeds easily is completely not understood. So there's a little strike through, it's not parallel. But there are lots of other traits that we can explore to understand, to explore the extent that parallelism exists between these species. I'm going to just briefly talk about some of the earliest domestication traits that we would expect to see, changes to plant architecture. So whether they'll grow in a way that sort of they spread out and take up a lot of space, or whether they'll grow really upright, and that way you can sort of produce more. You can increase the yield of what you would grow in your garden because you can grow more plants in the same region. So the most famous gene that controls this architecture, whether you're sort of splayed out or you're upright, is called PROG1, or prostate growth 1. And here is an image from Asian rice where the left two pictures are actually, so when you don't have PROG1 functioning properly, you get an upright plant. The wild type functional copy of PROG1 lets the plants be splayed out. So these are PROG1 mutants, so they don't have a functional PROG1, that have been transformed with a functional PROG1, and so their sort of spreading phenotype has recovered. And this is on the right is a wild type, non-functional PROG1 plant. Is that clear to people? Okay. So turns out that the wild progenitor of African rice and all African rice, except for the hybrids with Asian rice, none of them have this region at all. So we can't, there's no parallelism here. And it turns out all of them, even the wild ones, I did notice, even though they shattered, they all grew really upright. It was really easy to grow them in these really small containers in the greenhouse. That was my note to myself to tell you that. So there's some really surprising differences between these species, and this is a list of lots of different genes that have implications in domestication or diversification that I'm continuing